Hey everybody, welcome to the Brain Lab. This marks about halfway through our course, so congratulations getting this far. It is a hard course and you've survived this long. You got this. You can do the rest. Let's talk about this lab. So this lab looks at the nervous system and some specific aspects of it. Just put myself down there. So you're going to be looking at the brain, and there's a bunch of vocabulary to learn. Okay, so you need to learn where all of these features are in the brain. A lot of them especially happen within the cerebrum. So the cerebrum is what sets us apart from a lot of other animals. So you'll see that many of the brain parts, when you talk about lizard brain, you're talking about very basic parts of the brain. That's things like your pons, your medulla oblongata, things that just keep you alive. We'll talk a lot more about this in lecture. If you've ever heard the idea that you only use 10% of your brain, well, that's absolute nonsense. That comes from a horrible study in which scientists in the, I don't know, 40s, something like that, were like, let's look at this rat and let's just start stabbing its brain until it dies, and let's see, like, right before it dies, how much of its brain we were able to get rid of before it died. Now, people, that's not great. It's not a good study. The animals suffered unnecessarily, right? And so also what it found, think about, this was in a time where people didn't understand animal behavior, they didn't understand animal communication, all of these other things. So what they thought was a living animal just meant that it was breathing, its heart was beating. Now think about yourself. You are a much more complicated organism than a rat, and you do more than just breathe and have a beating heart. So to take any part of your brain away, or to think that any part of your brain is wasted, is nonsense. You use literally 100% of your brain. If not all the time, at least in a complete day. So some parts of your brain aren't active until you're sleeping, such as healing. Other parts of your brain are active at different parts in your life, such as like the parts that become active during pituitary or during puberty, such as those in the pituitary. But either way, you're using 100% of your brain, and so there's no limitless pill you can take and suddenly use more of your brain. That's silly nonsense. It's exciting science fiction, but it's not accurate anatomy. Okay, so after that rant is over, let's take a look at this. What do we got? We have bunches of parts of the brain and then parts of the cerebrum. Okay, so you're going to look and find where these are. So I would find a picture. There is a blank brain at the end of this, so you can use this to help guide your labeling, making sure you know what everything is, okay, or where everything is located. Remember, if you go find a lecture or a blank figure somewhere, you're only responsible for these different parts of the brain. Okay. Next, you're going to start looking at where the cerebrospinal fluid cycles. So this is the fluid system that feeds a lot of the brain. It also it does a lot of things. It helps cushion the brain during blows. It does a lot of very important things. Okay. There is a thought that the more plaque that built, and that's just like parts of dead cells, builds up in the cerebrospinal fluid, which is not only in the brain but also goes down to the spine, which is why it's the cerebrospinal fluid. And it also goes down through the spine. It's thought that the more of those plaques build up, that might be more likely to lead to Alzheimer's. Those plaques are cleaned up during sleep. Okay, so this part you want to look and see what is the shape of these, how do these things cycle, just kind of think, okay, so, and it'll walk you through it. So some of it starts here and then it moves here. So it'll walk you through what these ventricles look like and how they cycle that cerebrospinal fluid. You're then going to look at the meninges. There's 3 meninges. So if we've taken a man's skull and we have taken away the skin, this would be the skull. Okay, if we take a man's head, take away the skin, this is the skull. And then as you go down, you step downwards, there are these 3 layers that are between the skull and the brain, because the brain needs to be protected as much as possible. So you're trying to protect and make sure whatever is maybe something ends up getting through your skin and through your skull, can it get all the way through these 3 layers before it gets to the brain? Hopefully not. Something bad's going to happen. So you're going to look and find out which of these layers are which, and between each layer is a space. Okay, well, between 2 layers there's a space. Okay, so there's a subarachnoid space. Guess where that is? It's under the arachnoid. Okay, it's called the arachnoid because it's very, very thin, like a spider web. Okay. Okay, you're going to look at where the meninges are. If the word meningitis means anything to you, this is what gets inflamed during meningitis. One of these, if not more, will become inflamed. It can become inflamed due to a virus, due to a bacteria, due to a fungus, due to a parasite. There's so many ways that you can mess up the meningitis or the meninges because it's trying to protect the brain. Oh, okay, good stuff. Moving on. You're then going to go into this idea that it's very hard. The brain is so complicated, and we are brains trying to figure ourselves out, and it's an interesting question. Are we even smart enough as a species to really understand how the brain works? Well, we're getting there. We're trying to figure it out, but most of the time we only know if a part of the brain works because in someone else it's stopped working. Okay, so you're gonna get go through and look at like, so what's this part of the brain for? What's this part of the brain for? Where is speech held? Where is different kinds of things held? Right? What parts of the brains are responsible for other parts? And some of this will come from the lecture, so do look that up. You can also look in your textbook. It'll give you the figure number up here somewhere. In the textbook, it'll also go through these the functional sections of the brain. Okay. For example, our sight. So the brain, the way it's set up, the eyeballs would be here. But the part of our brain that processes sight is here. Why? Well, it's just how evolution set us up, right? So it's something to keep in mind that that's one of the reasons why, well, we'll get into it when we talk about the eye. It's very interesting. Okay. Part 5 is the neuron. So neural tissue, we talked about this very briefly in the tissue lab, in which I said, don't worry about this until we get to the neural tissue, or the neuron lab. And so now is that time where we should be able to look at a micrograph, so a microscopic photograph from a microscope of a neural tissue and be able to identify, okay, this is the cell body, so this means this must be this thing coming off it is the axon, this must be the nucleus, these other things are the dendrites, etc., etc. Okay, so it might help you to draw one, that might help you identify where things are. And lastly is the reflex arc. So the reflex arc, so if you ever touch something really hot and you move your hand really quickly, it happens before you even can think about it. It's literally happening before you can think about it. That thought process doesn't go through the brain. There's no thought involved at all. The reflex arc happens through the spine, and it happens you get a stimulus from a sensory neuron. It goes to your spine and says something happened, and then instead of taking the time to go to the brain, decide what to do, and come back, it automatically does the thing. And so you're going to look at these different kinds of reflexes. And in lab, usually when we're in person, we do these to each other. So we have these little reflex hammers, we have flashlights, we have different ways to do this. You can do some of these on people that you live with or your children, if you whatever you like. These are all safe as long as you do them correctly. Where it says a reflex hammer, please do not use a real hammer. Okay, only do this if you have a reflex hammer. I did provide you with, there are 2 labs to go. Oh, let's talk about this a little bit. You should be able to, when you're doing this, you want to look at, this is the reflex arc. So this is looking at a slice of spinal cord, and it's showing that, okay, if this is a neuron that comes in, well, what's this? Well, if this is coming in, well, what's happening? So you'll notice the loop stays within the spinal cord. It doesn't actually go to the brain. Okay, so there is a figure in your book that goes over this, so do look over that. You will need to know, again, these words, okay? And how to identify them. So that'll be important for the lab practical. Okay, so add these to your list of terms you need to know for this lab. And then to go with this, because we don't get to like poke and prod each other and look at reflexes, there are 2 simulations that go with this lab. One is on the pupillary test, where if you apply a certain amount of light to the pupil, it will reflexively, without even having to talk to the brain, will either get bigger or smaller depending on how much light is there. Now it turns out there is a way to cause this effect to happen that is not reflexive, and that is, a lot of times there's an emotional response that happens within the eyes, where if you're scared, your eyes get really big so that you can suddenly take in a lot, your pupils get really big, so you can take in as much light as possible to be able to test to figure out what's happening and get away from whatever's stressing you out. Okay, so you're gonna be looking at that. And then also, if you if you are around someone that you really like and you love them, your eyes also will get very big, or pupils will get very big to take in as much about them to keep processing. If there's something you really like, your eyes get really big. And this doesn't just have to be a person. This could be a thing, or an activity. When I was in high school, I was in the marching band, and I loved marching band. Imagine that. The professor was a big band nerd, and to the point where every time I would go to band to play my instrument, my pupils would get huge, and people asked me if I was on drugs. I was not on drugs. I was just very excited about playing my saxophone. Now I don't play the saxophone anymore, but I instead get excited about teaching. I still don't do drugs. Okay, all right, because anyway, we won't go down that path. So that's one of the labs. There is another one. and that is it's basically, so this, all of the reflexes we're looking at are called monosynaptic, which means there is one set of nerves that say, hey, do the thing, and that's it, they make it happen. That's as complicated as we need to be in this class. So in this one, you're going to be looking at, this is a reflex hammer, so it's going to show you exactly what happens when you have a reflex hammer. If you want, you can go look at Babinski reflex. You can go look at videos of other kinds of reflexes. It's very interesting. So babies have a different reflex when you tickle their foot than adults do. And if you see someone who has an adult foot but does the baby response, that means they have nerve damage. And so that's a good thing to know, because you need to figure out, oh no, well, if that nerve is damaged, what else is happening? What else could be happening? Okay, so you can look up those reflexes that are in these. If you aren't practicing them, you can look up the Achilles reflex, the patellar reflex, the plantar or Babinski reflex, the pupil, et cetera, to see, oh, what does that actually look like? And then read about it and then kind of get a better idea of what's going on. Interesting. Okay, so then you have your practice questions. There we go. Okay, you have your practice questions. If you can answer these from this lab, you understand the material in a very good way, and you can go on to the lab questions, the lab question set. So remember, these questions don't get turned in. They're just to help you practice and help you practice labeling. But then once you are done with them, you can go to the next thing. Let's see. Go. Okay, so then once you're done with that, you can go in and do the brain question set. This is mostly labeling and matching labels to where they go in the various structures, so do make sure you have your notes handy. Good luck on your Lecture 2 exam this week, and remember that Friday is a day off in observance of July 4th, so have a fabulous weekend. Make sure you get all of your assignments in before that time, and I will see you later. Goodbye.